Hello, my name is Matthew Tatz, and today I'll be doing a presentation on avian salmonellosis, a pathogen. My presentation title is A Survey of Avian Salmonellosis. This is for the Wildlife Disease Ecology course at the University of Georgia for fall 2020. Okay, so an overview of what we'll be looking at today, including an introduction to Salmonella enterica, what is this pathogen, history of Salmonella enterica, the epidemiology of avian salmonellosis, and then we'll be looking at that for passerines in particular, which is another word for feeder birds. And then we're going to look at an investigation of Salmonella enterica, including diagnostic tests, public health, and management. Okay, so what is Salmonella? First of all, Salmonella is a bacteria. It comes from the genus Salmonella. It looks like this. It has flagellum, which is how it moves, with the long, like, strings attached to it, I suppose you could say. It's a gram-negative bacilli bacteria. It has 2,300 serotypes, serotypes being subclassifications of this bacteria, which is quite a few. And it infects a diversity of hosts, from poultry to pigs, all sorts of kinds of birds, cows, and it does include people. Okay, so in this presentation, we're going to be focusing on avian salmonellosis. So avian, meaning we'll be taking a look at birds, and in particular, wild birds. And salmonellosis is the disease that Salmonella typhimurium enterica causes in the animal. Okay, so as I just said, Salmonella enterica, its serovar, one of those many serotypes I just mentioned, is typhimurium. It has over 300 different phage types, so that's another subclassification of this bacteria, which is really a lot. And in wild birds in particular, it seems that DT40 and DT56 are the two phage types that come up time and time again that are recirculating in wild bird populations. This disease is epizootic. Now, epizootic means that it appears seemingly at random locations. It doesn't occur necessarily in the same exact place at the same exact time every year. And this occurs in wild animals, or in this case, wild birds. Okay, so a little history about Salmonella typhimurium and avian salmonellosis in particular. It's considered an emerging disease. It's been noted that it increased exponentially over the past 40 years. This is due to artificial feeding. So in the past 40 years, there has been a surge in the amount of bird feeders people are putting out and other means of feeding wild birds, which causes the pathogen to transmit more easily. We'll get into detail on that soon. It was in 1957 that the first serotype typhimurium was even reported in wild birds, which is relatively recent. The researchers Hudson and Tudor found avian salmonellosis in quite a few different species: house sparrows, starlings, brown-headed cowbirds, and rusty blackbirds in northeastern US. Okay. So looking at the epidemiology of avian salmonellosis. So epidemiology is basically a review of breaking down this disease into its different components. So we're going to look at who does this disease occur in, what type of birds, where in particular are we noticing this disease to take place and spread, when is this happening in general, are there seasonal differences? Is this happening in certain times of the year? What exactly is this disease? And why is this disease happening? Why is it spreading? And what is going on here? Now, for the purposes of this presentation, we're going to be looking at the what first. So what is avian salmonellosis? That way you have a better idea of what it is when we go into the subsequent details. Okay. So first of all, what does avian salmonellosis do to birds? Well, in in disease ecology, we refer to clinical signs as observational detriments, I suppose you could say, to the animal, as opposed to symptoms, which would be self-reported in people. So there's actually quite a few clinical signs in birds for avian salmonellosis, and I'll run through them briefly. First of all, clinical signs, when you can notice that something's wrong with the bird, it's very brief, lasting only between 2 and 3 hours. So it's a very short window of time that you can actually see that this bird might have avian salmonellosis. They will be reluctant to fly and appear disoriented. This was noted in sparrows in particular. They'll often floof up their feathers and just remain very still. Then they'll collapse and die quickly. So not a very long period of time once it takes effect in the bird that you could notice it. So birds surviving more than 24 hours will fluff their feathers and be reluctant to move. So it's possible that they just die so quickly that you really don't even notice. Okay, so unlike Salmonella in people, which you might be commonly referred to as food poisoning, which often causes diarrhea, this is not a clinical sign in wild birds. The birds will have difficulty swallowing, so they'll have esophageal lesions. Here's a picture of an esophageal lesion, this cheese-like nodule in an evening grosbeak. So that causes inflammation, and the bird won't be able to eat as well, possibly. Weight loss is variable because this takes effect so quickly and leads to death so quickly. They might not even have time where perhaps their esophagus is swollen and they're not eating. They're not necessarily losing weight. They're just dying before anything, before they're starving to death, essentially. Salmonellosis takes place in the intestines, and so this can cause intestinal lesions in the bird. It also can cause enlarged intestinal and cloacal mucosa, so increased mucus in these areas. It can cause swollen liver, pale and enlarged spleens. It can have impaired blood coagulation, which can lead to hemorrhage into air spaces of the skull. And when looking at perhaps a dead bird, if you wanted to see if it had salmonella, Salmonella, the organisms themselves, can be isolated in large numbers from most body organs, including the crop, intestine, liver, myocardium, and the spleen. Okay, what are some notable birds revolving around avian salmonellosis? So we have raptors, otherwise referred to as birds of prey. So since salmonella takes place in the intestines, when these birds consume perhaps another smaller bird, and they're consuming parts of their intestine, they are directly contracting the Salmonella typhimurium from that small bird, as you can see there. So that's like a direct way to get it. Gulls, like in seagulls, their populations have increased greatly over the past 30 years. This is due to, again, anthropogenic feeding, anthropogenic meaning caused by people. And seagulls, like on the beach or in other areas like that, they'll go through trash, they'll take handouts, and they congregate so much that this causes Salmonella typhimurium, or ST, as I'll refer it from here on out, to spread in their population rather quickly. Pigeons. Pigeons, very similar case. Eating urban food, gathering in flocks. ST is spread through feces, which I'll get into, and so when they're gathering together, they're spreading this pathogen more quickly. And in general, urban foragers. So again, it's birds that might be going through the trash who are congregating in similar areas and are increasing transmission of the pathogen by being around each other, or just it's on the food in general. Okay, so in particular, we'll be looking at passerine for this presentation. So passerines is another way to say songbird. a more scientific way, if you will. They include any bird of the order Passeriformes, and this does include greater than half of all bird species. The picture on the right depicts a Carolina chickadee, which is a common songbird you might be familiar with. Now, what age of songbird does it impact particularly more than another? Well, in general, as with wildlife disease, younger animals or individuals are impacted more severely. This is especially true with Salmonella typhimurium because their intestines might not be fully developed, and so it's easier for the pathogen to take hold and cause problems. So there you have it. Okay, so is there a difference in passerines in sick versus healthy songbirds, or is there not? Well, first of all, it's important to note that a lot of the research that looks at if dead birds had Salmonella and Salmonella typhimurium, and if they're trying to figure out if it was avian salmonellosis that they died for, this data, when comparing it to healthy birds, is often going to be skewed because it's those dead birds that people are noticing and submitting for diagnostics that they're reviewing. So they're not going to be reviewing nearly as many healthy birds for Salmonella typhimurium, and so that's just going to skew data in and of itself. There were two researchers, Wolbeser and Finlinson, and they were looking at sparrows, and they found ST in 90% of the sick or dead sparrows, which is quite a few. But on the other hand, there was three other researchers, Izzard, Fish, and Harmison, and they reviewed the same population of house sparrows. Well, it might have been a different population, but they also reviewed house sparrows, and they isolated ST in only 15% of the healthy sparrows. So in this situation, it does look like sick or dead songbirds are being impacted more greatly, but that makes sense because they're the ones who are infected with it. Now, it should be noted that healthy songbirds can still contract ST and develop avian salmonellosis. It's not just sick birds that can develop the disease if they're infected. Okay, so where is this happening? So congregating birds. Okay, so Salmonella is passed through feces, and as such, when there are a congregation of birds together, the amount of feces that they're creating is going to be larger, and the amount of contact with that feces individuals will make will also be higher. So in turn, this is going to cause Salmonella to be transmitted at a much more higher rate than if birds were not gathering into one location. So examples of this include hooded cranes in Izumi, Japan. So the hooded crane is a species of bird that every winter they gather together in Izumi, Japan. About 70% of the entire species gathers in one place, and researchers were obviously concerned that there would be transmission of this pathogen since they were grouping so closely together, and they did find some ST in their population. Another example includes black skimmers, which is, so they're ground nesting birds, and they pack together in dense colonies. And so again, researchers were concerned that these birds were packing themselves so closely together that they would be spreading ST, and as such, it was the case. And then bird feeders is a enormous impact on congregation, and we'll go into that into more detail right now. So bird feeders. When you place out a bird feeder, you're obviously causing birds, songbirds, to come and congregate into one single area to get the resource, in this case bird seeds, and as such they are pooping at the bird feeder, and the poop stays in the bird feeder for a period of time, especially if you're not cleaning it. And so when other birds come to the bird feeder, they are very closely coming into contact with that feces, Also, if you have feces that, or if you have, well, first of all, yes, if you have birds that come and they poop on the ground, now you have feces on the ground. And then you might have squirrels or chipmunks or just other birds in general that are pushing seeds onto the floor. And now this seed is covered in feces, and there are ground birds who will come pick up the seeds, and or just these birds as well. Once the feeder starts running empty, and they're going to be contracting ST that way. So this sort of feeds the purpose of one evolutionary advantage that songbirds have. So it's the evolutionary advantage of poop and go. So birds, typically, they'll be flying and they'll poop and they'll deposit their feces somewhere, and they'll just keep on flying, and they're never going to come back into contact with that again, nor other individual, or it's not likely that other individuals of the species will also come into contact with that feces. But by causing the songbirds to pack into such tight areas and come close together, all of that feces is building up into one location, and the individuals are returning to that bird feeder over and over again, which is not typical for a wild bird to do. And so obviously transmission is a lot higher. And so as such, in a way, the greater amount of feed you have at your bird feeder is going to lead to a greater amount of death, because the more feed you have, the more birds are congregating at that feeder, the more feces is being produced, and the more transmission of the pathogen is happening. Okay, so in terms of temporal scale, this study was looking at ST in bird populations across England. And as you can see, as you can see in the graph, green and yellow dots both indicate sites where ST was isolated in an individual. And these dots are widespread across the entire country. And so ST is not just limited to one defined area, it's very widespread. Similarly, here's another researcher, Sonia Hernandez, who isolated ST outbreaks across America. And at least on the East Coast, you can see that there's many pockets of areas that ST was transmitted, and it goes all along the coast. So again, very widespread. Furthermore, Sonia, she also did research looking at if ST transmission was higher in different habitats. So looking at developed like urban habitats versus natural habitat. These graphs are showing, so Salmonella prevalence being on the y-axis to the left, and the different type of habitat on the x-axis. Here you can see Salmonella prevalence decreases as wetland cover increases, which is, this study was done for the white ibis, which is a wading bird, and they've started becoming urbanized. And basically she was seeing if Salmonella prevalence was higher in natural versus urban areas. And so here you see wetland, their natural area, as there was more of that, Salmonella prevalence was lower. Similarly, in barren herbaceous grassland, Salmonella prevalence was lower as that habitat space increased. Now, if you look at the graph at the bottom, this is looking at open developed land, so like urban areas. And as the land became more developed, more urbanized, if you will, Salmonella prevalence actually increased. And there's quite a few reasons for that, such as urban birds, especially the white ibis, have started begging for food, and so they're congregating in these small areas like parks, and they're coming back to these areas where people are, and so as such they are transmitting the pathogen Salmonella to each other. So when is this happening? So there's a couple instances I'd like to point out, a couple research experiments that were done, or okay, so yeah, a couple. There was multiple research done over outbreaks of salmonellosis, and they all occurred in the winter. So in the winter of '92 to '93, This was over a variety of species: pine siskins, evening grosbeak, sparrows, finches. There was unusually high rain and snow that year, and this ended in an outbreak. We'll talk about some reasons why that might be, but generally it's because, well, high rain and snow means less resources for the birds, so they were congregating at these feeders and relying on them more, and unusually high rain is stressful for the birds, and that depletes their energy reserves, so they were gathering more often. In the winter of 97 to 98, there was 73 songbird or passerine cases reported. In the prior 10 years, there was only 2 or 3 cases reported per year. So as you can see, there was an outbreak that happened due to the increased amount of bird feeders that people are providing in recent years. And the winter of 99 to 2000 in Sweden, this was an interesting example. So it was a cold, rainy summer, and farmers were growing linseed, but because it was cold and rainy, they were only able to harvest 20% of that stock. So only 20% of 32,000 acres of linseed was harvested. So the birds there, the songbirds, were able to live off of that feed for most of the summer and the fall, and their population became very large. And so an abnormally large amount of birds were able to survive autumn and early winter. But now that the population was so large and winter came, there was not nearly enough resources or food for the birds to forage, and so they were depending heavily on garden feeders. And so later in that winter, when all of these swarms of birds were congregating, ST was transmitted, and a lot of these birds developed avian salmonellosis, and the population really crashed due to this outbreak. And then in the winter of 06 and 08 and 09 in Hokkaido, Japan, another interesting example, there was a mass mortality event, this time involving Eurasian tree sparrows. They found, in 2008 and 2009, they found 202 deaths in 100 incidents at 94 study sites, which is quite a lot. At these study sites, 11 of 13 that were, of these sites, 11 of 13 were related to wild bird feeding, or 85%, which is quite a few. And they looked at sparrow fecal samples, and 6 of 8 of them had Salmonella typhimurium, 88% of that being the phage type DT40. Oh, and then this is covered here, but let me move that. I will also remind you that the event of the hood and crades I mentioned earlier at Izumi, Japan, how they all congregate, that also is happening in the winter, is when they congregate. Okay, so here are some graphs sort of illustrating that researchers have found this is occurring mostly in the winter as well. The graph on the left, this is the one I was talking about with the tree sparrows in Hokkaido, Japan. In January, light gray being carcasses and black being death-related incidents in these birds. The amount of carcasses and death-related incidents sort of spiked in January. So December, this is like early winter. Now later, mid sort of winter, we've got January, there's a spike. Later into winter, now February, just a very large amount of death in these sparrows congregating at feeders. And then it sort of tapers off as the winter ends. But as you can see, clearly in winter. Similarly, Dr. Hernandez, or Sonia Hernandez, she did some research looking at some birds, and similarly, you can see in her data, there was an outbreak that spiked in February, the same month as this graph over here, and it sort of tapered down as well, light gray being birds and the dark gray actually being outbreak in people. We're going to look at that example in a little while. So why is this happening? Contamination. So I've mentioned this sort of throughout the presentation, but Salmonella ST is transferred through feces or ingestion. So at these bird feeders, the birds are congregating, they're pooping everywhere, the feed is now covered in feces that the birds are eating, and they're contracting the pathogen that way. Now, this can also happen in contaminated water. So if you think about bird baths, if you're not cleaning your bird bath and an infected bird goes in the bird bath and then another bird comes, the pathogen will live in there too. And if the bird drinks some of that water, they can contract it that way. This also occurs to birds that live in very contaminated water, like waterfowl, perhaps. So some environmental factors that impact salmonellosis. So climate change, very interesting example. So climate change, of course, is causing a general warming every year. And as such, birds that basically once the, oh gosh, the word's escaping me, migration. So the birds are migrating as it starts to become winter. They start migrating into more up north where it's warmer. They're sort of chasing the heat and in order to find more food resources. And as you can see, this dark orange bar shows over the years the amount of distance that birds are traveling or migrating north towards the summer. Now, the problem that happens here is more birds are traveling northwards, so they're congregating more, and winter, due to global warming, is starting earlier in some places, and so that's causing birds to get there, and it's colder than they were expecting because it didn't used to turn winter so early, and so basically they have to rely again on bird feeders and things of such, and that causes a lot of crowding. So food availability, if there was low food availability in general, like in the winter, of course, they're relying on feeders and they are congregating. Or in the example I talked before about the linseed, if there's high food availability earlier in the year, the population spikes, and just by having a larger population, there's going to be higher rates of congregation of these birds in and around feeders, which is going to cause transmission levels to be higher, especially in the winter where food resources are low. Another environmental factor to consider is stress. So things such as rain or cold causes stress on the birds, and basically when the bird is stressed, if they're infected with ST, which doesn't necessarily mean that it's developed into avian salmonellosis, something like stress can cause it to develop into salmonellosis, which is problematic for the birds. And then things like deforestation and urbanization. So as we saw with Dr. Hernandez's graph prior, more urban areas, you're causing birds to congregate more because there's less habitat for them to be in, and then that causes more exposure to feces and etc. So in particular, seasonality, so bird feeder crowding, so especially in the winter when resources are low, they're all coming together at these bird feeders and they're relying on this single source. Bird feeders in the winter are a huge energy source, especially when we provide them with feeds such as peanuts, which is very high energy. The birds become reliant on this, and yeah, as that causes pathogen transmission, which is problematic. Okay, so physiological stressors of winter. In the winter, it's colder, and the birds basically have to spend more energy using the shorter hours of the day to collect as much food as they can in order to store fat in their bodies to keep them warm. And then they also have to devote some energy to sort of fluffing their feathers so that they can insulate heat. And that all of that energy requirement requires them to be eating more food, and there's less food available. So when there's a bird feeder there and it's providing high energy food like peanuts, birds are going to become readily dependent on these bird feeders. So there's some criteria for transmission of ST at bird feeders. In general, you need to have a species which has an adapted phage type of ST. So there are some more common species. In this case, we're talking about passerines or songbirds. You need to have a dense population of bird. That way, feces is sort of developing and the birds are congregating, and that transmission is happening more readily. And then stress factor, so such as the winter, where it's colder and they're devoting more energy to staying warm and not freezing to death, or perhaps there's a bad storm or other things. So another factor involving bird feeders is when you have just one bird feeder, that's causing a lot of birds to congregate, first of all, at one feeder, which is going to basically create a large amount of feces buildup in that one area. It also causes basically congregations is larger when you have just one feeder. Now, if you were to have more than one feeder, that would cause birds to compete less. And in that case, the stress would be lower, because at one bird feeder you have a lot of stress, which is contributing towards the development of infected birds to have the disease of salmonellosis. And if you have more bird feeders, you have less stress, and you're going to have less species build up in one location. So why? Okay, yes. So why is this happening? So reservoirs. Reservoirs meaning typically with some diseases, there's types of birds that do not get infected by the disease, but they get infected by the bacteria, so Salmonella in this case, but they don't develop into disease. It doesn't develop into disease. So in this case, they don't develop Salmonellosis, but they're still able to transmit the pathogen to other birds and get them infected. So some research talks about farm birds. Farm birds seem like a good example of what might be a reservoir species for Salmonellosis, because they stay around obviously farm animals, which can have Salmonella, and they're being exposed to a lot of feces right there, and so that's how they might pick it up. So their diet of seeds and insects is something that I found really interesting here. Half of their diet, so farm birds, half of their diet is in, seeds and insects make up half their diet in the summer, but in the winter it makes up more than 90%. So they're congregating a lot and picking up ST there, or so that's what we thought, and that's why researchers think in general they may be a good reservoir, but there has not been a study that shows that this is the case. There's no clear reservoir for ST. Nothing has been significantly shown yet. It's more of just speculation. And this is especially true, so to find if a species was a true reservoir, there would need to be there would need to be a high prevalence, so there would need to be a lot of Salmonella in a particular species all throughout the year, not just during outbreaks. But what we have found when looking at species so far is it's really a low prevalence in many species outside of an outbreak. So really just 2 or 3 birds out of hundreds of birds may be shown to have ST. So that's showing they're not really a clear reservoir. So a quick look at the pathology of Salmonellosis. So the Salmonella, Typhimurium, enterica, these bacterial cells, as you can see in blue, they attach to the intestinal epithelium, which is the tissue. It's tissue of the intestine. Then the bacteria invades the cells, as seen in the red circles. They start to invade the epithelial cells. Or alternatively, right here, this is a dendritic cell, and it'll grab that bacteria and pull it straight through into the submucosa. Sorry. Salmonella, so if it doesn't do that, the Salmonella replicates inside of the cytoplasm of the cell here. So it goes from 1 to 2. And then these bacteria are released into the submucosa. So they end up here. After that, the bacteria are internalized within phagocytes, as seen here, where they are able to basically leave the intestine, they disseminate through the lymph, and that gets into the bloodstream, which then causes infection in the birds. Okay, so now we're going to look at an investigation of avian salmonellosis. We're going to look at diagnostic tests. So how do we know if a bird has ST in them or they developed salmonellosis, and how do we type that? Public health in general involving ST, and then management of this disease. So diagnostic tests. Okay, so what are we doing before? How do we know if a bird is infected with ST and maybe have developed salmonellosis? So we take a blood sample of the bird and we put it on an agar plate as shown here. It's called bacterial culture. It takes 4 to 7 days. This is a relatively laborious method, but a benefit to it is it creates substrate, which are these little pockets of bacteria here that you can do tests on, such as DNA sequencing. And it's a high sensitivity test. So because you're able to test the substrate directly, sensitivity refers to when the individual that you sampled that has like salmonellosis, your test shows that yes, they are positive for salmonellosis. So it has a high sensitivity. On the other hand, there's a newer test, PCR, you might be familiar with, polymerase chain reaction. It's a lot faster. It takes 1 to 2 days. It's relatively less laborious. However, it has lower sensitivity, and this is due to the feces. Oh, I'm sorry, I said that blood was the sample you were taking. It's actually feces that you're taking the sample from. My apologies. So feces, the composition of it basically can inhibit, it acts as an inhibitor in PCR tests, so it can be difficult to discern what exactly you're looking at there. So in this picture, as you can see, this might be a feces sample in PCR, and it has all these different strains of bacteria it could possibly be. And then here circled in red, there's typhimurium, and you can sort of gauge how much that is. However, in PCR, you're not getting that substrate, so you're not able to do extra tests on it. And so for that reason, the gold standard, or the test to do, is bacterial culture. Okay, so diagnostic tests. So how are we able to tell what like phage type or serotype we're looking at? So for serotyping, before in America, generally people, they do what's called serotyping. They, on a plate, they create 2 wax circles. They put on a visible amount of bacteria on in both circles, and then they add a Salmonella antiserum, basically testing to see if the bacteria is indeed Salmonella, and if it clumps, then it is indeed positive. Okay, in Europe, they did phage typing, which is where you have a plate and you put on the bacteria into 4 different quadrants of the plate, and you're testing for specific phage types of the bacteria, and then you're able to discern if it has a particular phage type that you're looking for. So this is a more particular test. But now we just do genome sequencing, which is, it gets you exactly what you're looking for much quicker by breaking down the DNA. So diagnostic tests in more detail. So to look at the Salmonella in more detail, even more detail than like serotyping, you're going to do something, well, this is the gold standard for molecular subtyping. It's called macro restriction pulse field gel electrophoresis, or PFGE. It's a fine scale strain discrimination test. It basically separates DNA fragments by subjecting the gel to electrical currents, and there's a microbiologist doing this test. So that is the test to use. So here's an instance of a researcher that used PFGE on 50 different, or 1,477 different birds of 50 different species. In this, they were able to confirm salmonellosis in 263 adult birds, and they were able to get this down into the phage types of DT40 and DT56, which I referenced are the two most common ones. And you can see out of all the other phage types they got out of Salmonella, these were clearly the two most prevalent over these years. And they actually were able to find 90% genetic similarity between these two phage types as well. So here's another PFGE test that a researcher did. This was actually Sonia Hernandez again. So in 2009, there was a salmonellosis outbreak in humans involving peanuts. People were concerned that these peanuts might have ended up in bird feed and would transmit the pathogen into birds. So there was a bit of speculation about that. So Sonia's team, they surveyed the birds, so there was speculation that might have happened. At the same time, there was a species of bird called pine siskins, and an epizootic happened, which is there was an outbreak, and that's why people correlated that perhaps this human outbreak was related to the bird outbreak, and maybe they were related. So Sonia's team used PFGE to sort of break it down into very specific phage types, and so they were able to find that 94% of the cases in pine siskins and American goldfinch were here in A3. So, A3 right here. So if you go over here, you can see N being the sample size. 55 of those birds were related to phage type A3, clonal group A3. The other two high groups were A4 right here, and that's A6 and A4. And these were not the same clonal groups as determined in the human outbreak, so they were not the same. So another more specific test is, this is another layer down, even past the PFGE. This is multiple locus variable number tandem repeat analysis, or MLVA. So this is looking for genetic relatedness among those isolates that were found in PFGE. So in that same group, they were then able to take A3 and split it into two main groups. So A3A and A3B, seen down here. And basically, if you look at A3B, they found, and I'll zoom in on this, they found that 76% of the isolates in A3 were coming from a single strain. And this is the strain that was causing the problem. And if you look, which is interesting, this was occurring in North Carolina, Georgia, Tennessee, South Carolina, Virginia, Pennsylvania. So the same strain was found in all these different states, showing how widespread ST can be. And there you go. To put this map back into perspective, this is where that strain was found in this map. So in regard to public health, is salmonellosis a problem in wild birds? Can it get people sick? Yes, so salmonellosis can be a problem. It is a potential health concern for humans. In 1968, there was an outbreak in finches. There was another feeder outbreak. They actually, in this case, did find that it was the same phage type in humans and birds. So there was clearly contamination there. So to avoid that, you have to make sure you're being sanitary, when handling your bird feeders, so you don't pick anything like that up. And okay, so as we see here, so there was a researcher who in 1966 to 1996, so over 30 years, they were analyzing ST outbreaks in humans. This was in Norway. And they found that 78% of cases were occurring January to April, which was in the winter, which is quite interesting. And again, that is, here was Sonia's data involving the pine siskins. And you see that in winter, we talked about how in February and March, that's when the outbreak happened in the birds. Well, I mentioned earlier that the dark gray bars were humans. As you can see, the human outbreaks kind of spiked after this wild bird outbreak. So it's possible that that could be related in some way. So how to stay healthy? So if you're handling wild birds, or you're cleaning your bird feeder, or there's a dirty bird feeder or a bird bath that you're wanting to clean, obviously wash your hands. Don't eat or drink anything while handling animals or after cleaning your bird feeder, so that you don't contract it as well. And you can even wear gloves if you want to take the extra measure. So management, how to reduce contamination in general. So here are some ways to maintain your bird feeder so that you reduce the likelihood of transmission. You can limit the amount of feed that you're actually putting in your bird feeder. So if you're putting less feed, that's going to run through quickly. So birds aren't going to come to the feeder when there's no bird feed there. And so by putting less bird feed, the amount of time that birds are congregating at the feeder is going to be lower. So transmission would be lower. Keeping your feeders clean. If you see fecal matter building up over time, definitely clean it to keep transmissions low. Using bird feeders with smaller platforms. So birds are contracting feces. A lot of it is sitting on the platforms of the bird feeders, and the birds are coming to the feed and then contracting it that way. If you have a good feeder that doesn't get feed on the floor and doesn't really have a huge platform, like a small platform as shown in this picture here, then the poop will go on the floor and there won't be feed on the floor. So ideally, the transmission rate would be lower. And then providing several bird feeders. So as I mentioned earlier, instead of having one bird feeder where all the birds are competing and it's stressful and they're transmitting the disease due to contamination of the feces, by providing multiple bird feeders, you're reducing stress and you're reducing the amount of congregation, and also cleaning your bird bath. Okay, so in terms of like waste disposal, so the white ibis that I mentioned earlier, it's a wading bird, and similar to the Canada goose, it's now begging for food, and it's taking handouts. Now they will also go get into trash and stuff where Salmonella, basically the birds are going to visit these receptacles and they'll poop there, almost like it's one giant bird feeder. And so ways to reduce, ways to prevent that from happening is making sure that the birds can't access the waste. So making sure waste is well contained is one main way. And then making sure that the birds can't get in there. So in summary, be mindful when utilizing bird feeders. I put together an acronym to help you remember just in general about avian salmonellosis. So consider the 3 C's. Cleanliness, that involves making sure your bird feeders are clean and also making sure that you are cleansly when you are cleaning the bird feeders and, you know, not eating food or touching your mouth or face after doing that. And like I said, keeping the bird feeder clean so transmission's low. As seen here, you can sort of see the fecal contamination building up. Crowding, so you want to use multiple bird feeders so that crowding or congregation is lower, so stress is lower and there's not as much fecal buildup. And condition, so what is the condition? What season are you in? Is it winter? Are birds going to be heavily reliant on your bird feeder? You should be extra cautious in the winter if you're going to be putting out a bird feeder, because the birds are going to become heavily dependent on this, and that's going to cause transmission rates to go up. So just in general, some questions that you can consider. Who's accountable for this? So are we as people accountable for providing bird feeders? Should we be the ones to make sure that the transmission is as low as possible? Does this even matter? So does it matter that certain birds are dying and coming into having large drops in population? Thinking about, does this affect specialist species, or does this affect like species of concerns perhaps? Which I would imagine it does. Well, it does. And should we provide feed in the winter at all? So should we be helping out the wild birds by feeding them when resources are low, or are we causing more harm than good by having these outbreaks of avian salmonellosis? So thank you very much for listening to my presentation. I hope you found it informative. If you have any questions, you can email me at Matthew, M-A-T-T-H-E-W, period, Tatz, T-A-T-Z, @gmail.com. Thank you so much.